I'm glad to present to you our next presenter, Curtis, who will be presenting on multi-omics for human microbiome epidemiology and bioactive discovery. Curtis, if you are ready, kindly share your screen.
Great, thanks so much. And hopefully that's working.
Yes, it is. Thank you.
Well, thanks so much for the invitation to join, for putting together such a really interesting forum. And I will aim to very quickly today talk about some of the work that we do at the Harvard School of Public Health and our Microbiome Center on what I think of as microbiome epidemiology. So I'll speak at the end. A lot of what we focus on is providing resources, both computational and physical, for human microbiome population studies, which look a lot like other types of genetic or molecular epidemiology. What we'd like to be able to do is measure different parts of the human microbiome in different molecular ways, couple that with other types of molecular and epidemiological information, process our each slice of microbiome data in sort of a rational manner so that we can see microbes and microbial transcripts and gene products and metabolites, each in a controlled, well quality controlled way. Perform statistical analyses to associate those microbiome features with environmental exposures, health outcomes, meta-analyze that consistently within and across different populations, and ultimately be able to link human health outcomes that start from our big population studies back down to very specific molecular and microbial mechanisms that we can validate in controlled settings in mice or in the lab. So with this pattern of microbiome epidemiology, again, we try to provide resources, physical platforms, computational and analysis platforms to carry out this kind of work. One of the platforms, one of the resources that I'll talk about today is called the Human Microbiome Bioactives Resource, or HMBR. And it's a collaboration between our Microbiome Center, the University of Maryland Institute for Genome Sciences with Owen White and Anoop Marakar, Ronak Xavier at the Broad, and then Wendy Garrett and I co-direct the Harvard Microbiome Center. So you'll see Wendy pop up a few times in addition to her talk here yesterday. So what the Human Microbiome Bioactives Resource, or HMBR, does is look for bioactives of different types that are potentially produced by the microbiome. And we define bioactives fairly broadly, such that they can be whole microbes that are resident and cause a health outcome or a phenotype of some sort. Direct mechanisms, such as microbial production of chemicals that go on to have a bioactivity, or indirect mechanisms. So microbial production of gene products, for example, that interact with human cells or the human immunology that go on to cause a phenotype. So the HMBR includes many different components for exploring different types of bioactivity, all the way from collecting physical samples, analyzing them individually, up through predicting different types of these bioactives, screening them in the lab, and making them available online. I won't have time to talk about much of this today. I'll highlight a few pieces from the middle here, where we're finding ways to predict and prioritize different potential bioactive products from the microbiome. And anytime you see a URL on these slides, such as the HMBR website down at the bottom here, there's always more information available online. So we've been developing, as part of the HMBR, a couple different systems for predicting different types of potential bioactivity from the microbiome. And one such is chemical or small molecule bioactivity. So whenever we look at small molecules, metabolites associated with different human microbiome body sites, everyone likes to talk about dark matter in the microbiome and how many microbes we don't recognize. There are how many microbial genes we don't recognize. There are whole bunches of microbial metabolites that we don't recognize. So if we look, for example, at an example of a collection of stool metabolomes, depending on how you measure, a typical stool sample can have anywhere from 40 to 80,000 individual chemical features in it, using untargeted mass spec-based metabolomics. Only a few hundred to generously 1,000 of those metabolites are typically identifiable. The vast majority of those metabolites aren't yet known and thus might be sources of bioactivity. They might cause some phenotype upon interacting with the human host. There's been some great work using tandem mass spectral methods to explore these uncharacterized metabolites, especially using guilt by association. So given an MS-MS spectrum of a new compound, if it looks kind of like the MS-MS spectrum of a known compound, then they're probably related somehow. And this provides a type of guilt by association that works great when you're able to have this type of complex data. More commonly, we have simple single mass spec data, or NMR metabolomics data, in which all we really know is the abundance of these unknown metabolites, but we can still search for guilt by association by looking at their co-occurrence or co-variation with characterized metabolites. So we can try to guess what unknown metabolites are by looking which known metabolites they co-occur with. This has turned out to work really well in a computational system called Macron, or Metabolome Analysis and Combined Annotation Ranks of Prediction of Novel Bioactives, which is why we give it a shorter name. What Macron does is group together known and unknown metabolites from the microbiome into modules and use guilt by association to transfer potential annotations to potentially bioactive molecules of interest. So if we use the same example from a few slides ago of stool metabolomes from the Human Microbiome Project, which comprise control individuals and inflammatory bowel disease patients, Crohn's disease and ulcerative colitis patients, these start out with several tens of thousands of features of metabolites, only a few hundred of which are annotated. Macron clusters those 40-odd thousand metabolites together into just over 600 modules, 150 of which contain at least one known metabolite. There's about 14,000 compounds that are included in these modules and thus have an opportunity to transfer some information using guilt by association. So this tells us something about what those uncharacterized metabolites might be. In terms of figuring out which of these metabolites might be important, Macron also integrates information from biochemical features of the mass spec data, which compounds might have been modified by microbes, for example, epidemiological features, like which compounds associate with a phenotype of interest, like inflammation in the case of inflammatory bowel disease, and information from ecology, which compounds tend to occur a lot in tandem with microbes, for example. So this gives us an integrated prioritization of which compounds might be potentially relevant for human health by combining these features as ranks and ending up getting a ranked prioritization of all compounds. I won't have time to talk about a lot of the details of our results in inflammatory bowel disease today, but in this one data set, out of the 150 modules that Macron forms that contain some information, they tend to cluster into one or at most a couple of known metabolite classes, ranging from bile acids through urea derivatives, fatty acids, and so on and so on. And in a couple of cases, we've been able to go all the way through this pipeline to drill down on individual compounds where we have a prediction of what they might be. So in this particular module, there were a few copies of the same unknown compound related to nicotinamide that we predicted to be nicotinamide riboside or vitamin B3, which were implicated in Crohn's disease specifically, which turned out to actually be anti-inflammatory. We're looking into the ways in which microbes might be generating nicotinamide riboside and its derivatives from nicotinamide still. But in a mouse model of colon injury run in Wendy's lab, supplementation with nicotinamide riboside did indeed alleviate gut injury as a result of this DSS perturbation, which is exactly what we'd predict from the human phenotype as well. So we have at least one case out of these several hundred possible cases now in which we've been able to follow that entire process of starting from a large human population, prioritizing mechanisms, molecules of interest based on a human health phenotype, drilling down into some computational predictions about why that might be, and turning out to verify those in the lab. So I mentioned that there's a couple different types of bioactivity and prioritization within the HMBR. Another very similar system to Macron prioritizes microbial gene products with bioactivity instead. So this second pipeline, Metavibla, I won't bother with the long name here, works very similarly, but instead of starting with metabolomics, starts with shotgun metagenomics and explores novel microbial gene families, open reading frames, in collections of metagenomes, rather than novel uncharacterized metabolites in collections of metabolomes. Otherwise, the idea is quite similar. Metavibla starts with a collection of open reading frames, clusters them into first nucleotide and then protein families, assesses which of these protein families already look like known proteins versus having no homology and thus being really novel, and then also tries to prioritize them based on ecological and environmental and phenotypic properties into which ones might be potentially bioactive, very much like what Macron does. So a combination of prioritization of which of this dark matter might be interesting and important for human health, and some guilt by association techniques to make guesses about why that might be, what mechanisms are driving that importance. So again, I won't have time to talk about a lot of the details, but in another example application of Metavibla to the HMP2 dataset, again, inflammatory bowel disease patients and controls, these are really well-characterized human gut microbiomes, but even there's a surprising amount of uncharacterized proteins with potential bioactivity and relevance to human health. Out of about 45 million original open reading frames, in the 15 or 1600 metagenomes from this population, these cluster down to just over 2 million nucleotide gene families and slightly fewer, 1.6 million amino acid-based protein families, remarkably 70% of which, even in the well-studied human gut, are still uncharacterized, either because their sequences just don't look much like well-studied proteins, or because the sequences that are homologous to microbial isolate proteins haven't been characterized in those isolates yet. So this gives us a really rich resource of compounds to potentially mine for novel bioactivity. And again, in a few cases, we've had success so far in drilling down individual prioritized examples to mechanisms of interest. Some of these include, for example, differential pillin structures that are carried by Enterobacteriaceae as compared to Bacteroides in the human gut. So collections of mostly type 1 and T pillins were prioritized as inflammation associated in subsets of E. coli in the gut. These were able to be verified both by some independent high throughput data. So in terms of metatranscriptomics, these pillin families were uniquely overexpressed just during active dysbiosis, during inflammation and IBD. And I won't have time to talk about it today, but we also have some separate isolate-based microbial genetic evidence that this is true as well. So to wrap up, since I'm just about at time, the HMBR, the Human Microbiome Bioactives resource, is one of the resources offered by the Harvard Chan School's Microbiome and Public Health Center, which again, Wendy and I have launched together a few years ago, and we really try to push out resources for training, for students, for research, for outreach about the role of the microbiome in population health. So if you'd like to follow up on anything from the Microbiome Center, please take a look at our website. April Pollock is our center manager, and you're welcome to get in touch with her. And I'd really like to thank anyone whose face you saw on the slide are usually lab members who carried out some of this work. And I called out our collaborators on these projects already, the center members, and especially, of course, Wendy, who's both been great to work with on the center and participated in several of these research projects. So if there's time, I'm happy to take a few questions. Thanks, everyone.
Thank you, Curtis. We can have a couple of questions, right, Emma?
Yes, we can. We can have one or two.
So maybe I'll start with one. So you mentioned, so yesterday we were talking about how to define a healthy microbiota, and we were discussing this, and I think one of the tendencies is to say that we probably need to be focusing more on function instead of just names of microbes and category and species. And here you're focusing a lot on functions. But I guess the challenge is this huge unknown material of metabolites that is still out there, right? What's your opinion about how to progress in defining what's a healthy microbiota?
It's a great question, and we've had interesting conversations about that for a while now. Ever since some of the earlier exploration in how to combine function, define a healthy microbiome by combining functional modules. So I use the analogy of trying to build a working machine. There are lots of different ways that you can build a working ecology, but you have to have a collection of parts that work together and drive that machine. So you can select different parts. You can perhaps get the same functions from different microbes, for example, but one way or another, to have a healthy ecology, you have to build a working machine. What we run into with a lot of these uncharacterized function is that there are definitely parts, there are building blocks that are necessary. We don't know what they're doing yet. So sometimes we can recognize that in terms of, say, common functional modules, common enzymes, some of these prioritized enzymes, for example, that are probably important, probably bioactive. We just don't know what they're doing yet. So that gives a priority for new functions to study. Some of what I think we're missing that's probably really interesting is the long tail of functionality that's not common, that's not part of that sort of core functional healthy microbiome. There's probably quite a bit of personalization in microbiome function that we're not able to easily see yet because it's buried out there in the sort of uncharacterized dark matter. So there's been a couple of nice publications of examples of that. One that I really like is Peter Turnbaugh's work on microbial metabolism of digoxin, where they've done that same thing. They've followed a very nice workflow from defining a big human health phenotype of microbial inactivation of a drug, tracing that through to a very specific microbe with a very specific pathway that carries out that inactivation. Some people have that bug in that pathway, and some people don't. And so that's a case where there's sort of a little add-on module. It's an extra piece that's not part of a piece of functionality that's not part of the core microbiome that we wouldn't know about except for that really nice piece of work. And there's probably 1,000 or 10,000 examples like that out there that we still need to find. So both on the core health side and on the sort of personalization side, I get excited about exploring some of these uncharacterized functions.
Thank you. Emma, do you want to add something? I don't see any questions in the chat.
There's one question in the chat.
Okay.
It's from Pascal. She says, do you think the core functional microbiome is the same regardless of nutritional specificities? That is, do you think the core microbiome of a hunter-gatherer population is the same as in New York City?
Well, for a very extreme example, definitely not. But I would suspect it happens on a continuum. There's no sort of cliff that you'll fall off when in going from say one well-functioning microbiome to one completely broken microbiome or completely different healthy microbiome. But there could easily be a continuum across different configurations that are healthy in different environments. And we see that in at least Western diets that have been more deeply studied at this point, where long-term, the longer you have a dietary perturbation and the gradually more extreme it is, the more that sort of pushes the microbiome. So we see really stark contrasts between something like New York and a hunter-gatherer population where those are extremes of that continuum. But there should be, and again, there is, at least in the North American populations that have been studied, a continuum in between those along which the microbiome is sort of gradually responsive to diet.
Thank you very much. I think we're going to have to move on.